Hi, and welcome to Reviewing with Mrs. Wages. Today, let's review genetics. Let's get started with objective number 1, which asks you to prove allele dominance. We've got two fish here that are displaying very obviously different characteristics. What kind of an experiment would you conduct in order to determine which characteristic is dominant? Well, the answer, of course, is to do a P-generation cross, which means that we're going to crossbreed a purebred orange fish with a purebred blue fish. We know that they're purebreds, we don't know which allele is dominant. Every scientific experiment begins with a formalized hypothesis. In this case, that means an if, and, then, and because statement. In this formalized statement, the if is referring to your hypothesis, your simple hypothesis, what you think is the right answer. The and refers to the test you might conduct that would see if your hypothesis is supported or not. The then refers to the prediction of results for your test based on your hypothesis. And the because refers to your explanation of why you think this all should happen. Now let me show you an example of a formalized hypothesis that has nothing to do with genetics, but makes use of the same format. If fertilizer improves plant growth, that's my hypothesis, and I compare fertilized plants to non-fertilized plants, that's my test, then fertilized plants should be taller, that's my prediction, because they will get more minerals for cell production than the non-fertilized plants. That's my explanation. Pause the video and see if you can write a formalized hypothesis for this test, where we have a purebred orange fish crossed with a purebred blue fish, assuming that blue is dominant. Now compare what you wrote to what I would have written. If blue is the dominant allele, my hypothesis, and I crossbreed purebred blue and orange fish, my test, then all the offspring should be blue, my prediction, because they would inherit both alleles but only express the dominant one, my explanation. How did you do? Okay, before we put our hypothesis to the test, let's consider two very important vocabulary words in genetics. Those words are phenotype and genotype. What is a phenotype? A phenotype is the physical description of a trait. So then what's a genotype? A genotype is the combination of genes that controls that trait. So now let's see how to apply these words to our fish. Well, what's the phenotype of these fish? Well, that's easy. Just look at them. What's the difference? What's the phenotype of the fish on the left? This fish is orange, so its phenotype is orange. What about the fish on the right? What's its phenotype? This fish is blue, so its phenotype is blue. But it gets a little trickier when discussing genotypes. You've got to consider a few things before you can figure out what the genotype of each fish would be. Now remember, we don't really know which color is dominant, so we are making a hypothesis as to the genotype of each of these fish. And we also have to consider what generation that we're crossing. So we're suggesting that blue is dominant, and we know that we're crossing the P generation. With those two pieces of information now, we can hypothesize the genotype of each of these fish. Because we assume orange to be recessive, and we know this is a purebred, we can assume the genotype of this fish is homozygous recessive, or little b. Again, if blue is dominant, and this is a purebred fish on the right side, we can assume its genotype to be homozygous dominant, or big B. And now we're ready to plug all this information into a Punnett square. A lot of people incorrectly assume that a Punnett square provides results to a test. It does not. Punnett squares are only as good as your hypothesis because the information you plug into a Punnett square is actually based upon your hypothesis. So let's see how that statement relates to our actual experiment. So let's segregate the genes of our P generation parents on the outside of our Punnett square. Then we'll combine them together on the inside to show F1 results based on our hypothesis on the inside of the Punnett square. Then next we can take a look at the phenotype and genotype ratios from this Punnett square. Remember, inside the Punnett square is the F1, and based on this Punnett square, the phenotypes of the F1 should be 100% blue and 0% orange, because every single organism inherited that dominant blue gene, if we're right. In terms of genotypes, the F1 are predicted to be 100% heterozygous, 0% homozygous dominant, and 0% homozygous recessive. Now remember, all this data over here is based on our hypothesis, and we won't know if it's right until we actually look at the fish produced by our crossbreeding experiment. So we have to compare our predictions to the actual outcome of the fish. If we're right, the actual outcome should match pretty well with what we predicted in our hypothesis. Let's take a look. This is the actual outcome of our experiment. 100% of the F1 are orange, completely the opposite of what we suggested in our hypothesis. Was the hypothesis supported? No. Is that okay? Yeah, because we can still draw conclusions about this experiment. Pause the video now and see if you can write out a formalized conclusion based on this experiment, knowing that the F1 all came out orange. Be sure to address what the experiment was asking you to test. How'd you do? Compare your response to mine. I conclude that orange is the dominant allele because the F1 fish, which were orange, inherited both the blue and orange alleles, but they only express the orange one, proving that it must be dominant over blue. All right, on to objective number 2, which asks you to prove the masking of a recessive allele. To do this, we need a whole new experiment and a whole new formalized hypothesis. It may help to consider Gregor Mendel at this point. Remember, he crossed those purebred tall plants and those purebred short plants, and all of their offspring came out tall, and he was very confused. He didn't know if the tallness information was covering up the shortness information, or the short information was completely gone forever. So he decided to do an experiment. So how could you test whether a recessive allele is forever gone or simply being masked by the dominant allele in the F1 generation? The answer is an F1 cross. Let's take some orange F1 fish, cross them together, observe their offspring, and see if that blue phenotype reemerges in the F2 generation. So what's our simple hypothesis? Is the blue allele gone, or is it just being masked by the dominant orange allele? Let's assume that it's being masked. It's present, just being covered up by that dominant orange gene. Now it's time to take that simple hypothesis and write a formalized version. Remember, you need an if, and, then, and because statement to formalize this. Pause the video and write out your formalized hypothesis for this experiment, then compare your response to mine. If the F1 fish have a masked recessive allele, my hypothesis, and I cross orange F1 fish, my test, then 25% of the offspring should be blue, my prediction, because 1/4 of the F2 offspring are predicted to inherit a recessive allele from each of their parents. This would prove the presence of the recessive allele in the F1 generation. My explanation. Did your hypothesis have all the required components? Okay, let's produce some F2 outcomes using a Punnett square based on our hypothesis. Now remember, we're assuming that the orange F1 fish are hybrids, and they actually possess a masked recessive blue allele. So let's take the genotypes of our F1 fish and put them on the outside of the Punnett square, segregating their alleles, and looking at all the possible combinations of F2 fish on the inside of the Punnett square. And remember, Punnett squares are not results. They're just predictions, but we can use these predictions and compare them to the actual results of the living fish. So let's examine the ratios for both phenotypes and genotypes of the F2 based on our hypothesis. This Punnett square suggests that 75% of the F2 should be orange and 25% should be blue. These 3 out of 4 have the dominant allele, making them orange. This 1 out of 4 only has recessive alleles, making it blue. As for genotypes, 25% are suggested to be homozygous dominant, right here. 50% are to be heterozygous, these 2 out of 4. 25% are to be homozygous recessive, right over here. But again, these are just predictions. We have to look at the actual outcomes to get our final results. Our actual results show that 6 out of 8 fish are orange and 2 out of 8 fish are actually blue. This also means that 25% of the fish, or 2 out of the 8, should be homozygous dominant, and 50%, which is 4 out of 8, should be heterozygous. The last 25% should be homozygous recessive, which would be these blue fish. Was our hypothesis supported? Yes, it was. What can we conclude about the presence of the recessive gene in the F1 generation? Well, you tell me. Pause the video, write down a formalized conclusion, and then compare yours to mine. Here's what I concluded. 25% of the F2 expressed the recessive gene, which means that each of their parents gave them one recessive allele. This proves the recessive allele was present in the F1 but being masked by the dominant orange allele. Now, does your conclusion fully explain the details of your experiment and results? All right, it's time to flash back to glycolysis. You should know 3 general things about glycolysis. What is it? Where does it happen? And why does it occur? Let's begin with what. Glycolysis is the process that converts glucose into a net gain of 2 ATP molecules. It also makes pyruvate and NADH. Glycolysis occurs in the cytoplasm of cells. The ATP produced by glycolysis can be used for cellular activity. The pyruvate and the NADH, these are the reactants for both aerobic and anaerobic respiration. If oxygen is available, pyruvate and NADH go to the mitochondria and carry out aerobic respiration, which makes a whole lot of ATP, and it's the most efficient pathway. If oxygen is not available, pyruvate and NADH stay in the cytoplasm to carry out anaerobic respiration. Anaerobic respiration is also known as fermentation, whether producing lactic acid or alcohol. This pathway does not produce ATP at all. But the benefit of fermentation is that it does produce NAD positive. And as you remember, NAD positive can then be used in glycolysis to make a bit of ATP, getting us by until oxygen is more readily available for aerobic respiration. That's all for now. I hope this helped. And as always, happy studying!